1 second. I'm not doing this without a cup of tea. Not bad. Okay, so if we've understood the Krebs cycle, then it's time to take it a step further. The last thing we saw was this diagram. This is showing us where we're at. We've finished the Krebs cycle, we've got a little bit more ATP and all those other things that we made from our 3 stages so far. Now it's time to take this to the final stage where most of the ATP is made, and that is the electron transport chain, where we get oxidative phosphorylation going down. Let's have a look. Okay, so this is the diagram that shows you the electron transport chain and everything that happens in it. There's a lot going on in this diagram. What I'm going to do is sort of talk you through the overview of what's happening here first. Then I'm going to go briefly through what's happening, show the diagram again, and then go through it again, but with a little bit more detail. Triple whammy. So let's look at an overview of what's going on here. Look at the top of the image, look at the mitochondria, and see where we're at. We're zoomed in here on the membrane of the cristae, the folded membrane, the inner membrane. That's what this whole image is. So we're looking at proteins that are embedded within that inner membrane of the mitochondria, the inner folded membrane. Now we're going to read the diagram from left to right, okay? Look at what we're seeing. The darker orange bit at the top, that represents the space inside, between the inner and the outer membrane, right? We call it the intermembranal space. Then we've got the membrane itself. The lighter yellow represents the matrix, the actual space inside the mitochondria that makes up, most of the room where the Krebs cycle is happening. So, reading from left to right, what we can see is NADH arrives and donates something, right? You can see a yellow arrow coming out of NADH. It's donating 2 things. It's going to let go of the hydrogen that it's been carrying, and it's also going to donate an electron. So NADH is oxidized. It loses electrons and hydrogen. The electron is passed to the first protein in a chain of proteins in the membrane. That electron gets passed from one protein to the next. You don't need to know the names of any of these proteins except for one of them. You'll notice one of them, the 4th one in the chain, is labeled Cyt C. That's Cytochrome C. Cytochrome C comes up in multiple topics. It came up in Origin of Cells, it comes up here, it comes up again when we talk about evolution. Cytochrome C is one of the molecules that is what we call a universal molecule. It's found in all living things, and therefore we can compare its structure in all living things to identify how closely related we are to other organisms. So Cytochrome C is a molecule from multiple topics, therefore it gets a special mention here. But you don't need to know the names of the proteins in this chain, particularly it's not important, just understand that there are proteins in a chain fixed in the membrane. So the electron is passed from one protein to the next. Every time it does that, it releases some energy, and the energy is used by these proteins to pump the hydrogen that came from the NADH and all the hydrogen we've been producing all the way through this process through the proteins into the intermembranal space. The hydrogen ions, sometimes just call them protons, because essentially that's what they are. They're hydrogen without electrons is basically protons. Those hydrogen ions are pumped into the intermembranal space and they build up in concentration until they're extremely high in concentration in that space. The electron continues being passed between proteins until eventually it's delivered from the final protein to an oxygen atom. Oxygen picks up the electron. It's the final electron acceptor. It then binds with some spare hydrogen and forms water, H2O. Now, all that hydrogen that we've pumped through into the intermembranal space, it has to go somewhere. It's built up in concentration. There's now a huge concentration gradient between that space and what's on the other side of the membrane, which is the matrix. So that hydrogen is desperate to flow back down its concentration gradient. And it does that but the only way out is through a particularly special protein called ATP synthase. ATP synthase is a channel protein, but it's also an enzyme. As the ions flow through ATP synthase, the energy from that flow of hydrogen is utilized to phosphorylate ADP molecules. And that's what's producing all of the ATP. The more hydrogen that flows through it, the more it can phosphorylate ADP. It's called oxidative phosphorylation because if you don't have oxygen as that final electron acceptor, the electron won't even move down the chain. So nothing happens. You have to have oxygen there to make that electron transport chain happen. So you need oxygen to phosphorylate the ADP. Oxidative phosphorylation. Now let's go through this with a little bit more detail, then look at the diagram again, and then go again, and then look at the diagram again. The first step is oxidation of the reduced coenzymes. NAD and FAD are coenzymes. They've been reduced, which means they picked up hydrogen. They are now going to do the opposite of that. They're going to be oxidized. They're going to release their hydrogen and release electrons. The electrons are passed between membrane proteins. The membrane proteins pump protons into the space between the outer and the inner mitochondrial membrane. These hydrogen ions form a high concentration and, as a consequence, also a low pH within the intermembranous space. They then diffuse back down their concentration gradient to the matrix through a channel in the membrane, which is a protein called ATP synthase. The flow of hydrogen through ATP synthase drives an enzyme reaction that brings about the phosphorylation of ADP and some phosphate ions into ATP. Okay? Alright, let me give you some more solid notes. So these electrons are going to get donated by their electron carriers and pass along a chain. So what's literally happening is the NADH and the FADH2 are both oxidized, which means they lose electrons and hydrogen. Proteins pick up those electrons, pass them along a chain using energy to transport hydrogen from the matrix into the space between the two membranes of the mitochondria. That produces a big concentration gradient. The energy for the pumping of the hydrogen came from the NADH and the FADH2. So the more of that we have, the more hydrogen pumping we can do. The electrons flow along the proteins, reducing and then oxidizing each protein one by one as it's passed along. Those redox reactions release energy that can be used by the proteins to actually pump hydrogen. So that hydrogen builds up in the intermembranous space. Alright, so that's a form of active transport. Eventually, the electrons get to the last protein and then are passed from the last protein to oxygen, or rather oxygen attracts the electron from the last protein. In fact, oxygen has the highest affinity for the electrons of all, has the greatest pull. It's the reason the electrons move down the chain at all. It's the final electron acceptor, so oxygen takes that electron and becomes negatively charged, eventually therefore attracting some hydrogen, which is positively charged, and forming water. That super high concentration of hydrogen that we've built up in the intermembranous space diffuses down the concentration gradient through ATP synthase, which forms ATP from some ADP. How on earth does that happen? Well, it's actually quite remarkable the way it works. It's like little crazy nanomachines. The ion flow, the hydrogen flow, actually causes the head of the ATP synthase to rotate round and spin like a turbine, a bit like water flowing down through a water turbine and causing it to spin around. And that rotation actually collides ADP molecules with phosphate ions and causes them to react. So the more hydrogen that flows down that gradient, the more rotations, the more collisions, the more ATP. For every NADH molecule, you get 3 ATP molecules, and for every FADH2, slightly less, you get 2 ATP molecules. Now if you do the math, you can then work out how many ATP molecules in total are made by the whole respiration process. It's quite a lot. See if you can figure it out. And that's it. That's respiration, that's the electron transport chain. So the more NADH and the more FADH2 that we can make, the more hydrogen and electrons we can supply to our electron transport chain, the more hydrogen we can pump into the membrane, the more hydrogen will flow down through the ATP synthase, and the more ATP we can make. That's why we've been building up our NADH supply all the way through the process to this point. In total, 38 ATP molecules can be made. Most of that, the vast majority of it, is made in the electron transport chain. We got a few earlier on, but the majority came from this process, this oxidative phosphorylation that happens here. Remember, we don't keep 38 because some ATP is used by the mitochondria to pump in the pyruvate, and some of it is used at the beginning in glycolysis. Okay, hopefully that all makes sense. I've put a little table on the OneNote for you to fill in to try and figure out how many molecules of ATP and NADH and FADH2 and so on are made throughout the entire process. Try and fill that in, and then I've got a few questions for you guys to see if you get it. Good luck.